I'm Wen-Yao Lee from National Jiao University. Today, I'm going to presenting on the topic single-cell network-based approach to investigate intratumoral heterogeneity in progression of head and neck cancer. Firstly, head and neck squamous cell carcinoma is currently the sixth most common type of cancer in the world, and over 65% of the patients experience recurrent and metastasis, which leading the high mortality rate. And epithelial mesenchymal transition plays a crucial role in HNSCC metastasis. So it is important to emphasize that EMT contribute to poor prognosis in HNSCC. And although EMT may lead to the worst prognosis, it remains a reversible process and presenting a potential window for a therapeutic intervention. But most of traditional therapies is ineffective at removing the cancer stem cell that arise through the EMT progression. And most of them exhibit hybrid epithelial and mesenchymal state, which has strong capability for metastasis. In the past, bulk tissue research methods struggled to reveal the heterogeneity of cell subpopulation within the tumor. But recently, single-cell RNA-seq provided opportunity to explore intratumoral and cellular heterogeneity at single-cell resolution. Here, although single-cell RNA-seq provide a lot of information, but traditional methods such as differential expression analysis is hard to capture the gene-gene association, lacking the ability for understanding the system-level change in gene regulations. On the other hand, single-cell network biology using node and age representing the gene and their interaction provide a comprehensive understanding of the relationship between genes at the system level. Although there's a lot of method developed to construct the biological network, most of them using group of cells to construct the cell type aggregated network. These approaches are hard to reflect the cell-to-cell heterogeneity. So recently, the concept one cell for one network has been proposed, such as cell-specific network, CCSN, and SWEET. One of the most well-known methods is cell-specific network. It claims that if these two genes X and Y are correlated in this target cell K, a correlation must also be observable in other cells and clustered in the central box here, the central box. But the main problem of this model is its high false positive rate because the influence of the joint cell, which may include a lot of different cell type cells. It causes CSN suffers from an overestimation of age. Here, we want to propose a novel method called chi-square estimate of single-cell network, abbreviated as CHOSEN. We hypothesize that if the same cell association pattern should only be observed within the same cell cluster. So we incorporate with the transcriptomic correlation into our analysis, where the cell is more similar to the target cell. We will give it a higher weight. We hope this approach can denoise the influence of joint cell, which are different cell type. And then we are also the first one to consider the overall distribution of cell in this scatter plot by using chi-square estimate. So let's dive into our framework of CHOSEN. First, we construct the weight matrix by using Pearson correlation coefficient, and then we create a two-dimensional scatter plot for each gene pair in the target cell, okay? In the target cell. And then we incorporate the cellular similarity weight into our analysis to construct the contingency table and using chi-square test to determine the significant cell association. After that, we have in a single cell network, we can convert it into the network degree matrix, which reflect the importance of each gene in the network. Then let's move to the result. One of the most common analysis in single cell study is identification of cell types. So here we're using 10 single cell RNA data set and hire the Levin method as our clustering algorithm. First, we can see in dominance data set, it is really hard to cluster clearly in the gene expression level, but especially the cell type OPC in our chosen method, it is almost separated into individual cluster. And compared to other methods, most of them are completely mixture with other cell types making the identification quite challenging. There's also, we are also have outperformance for identification of cell types in other data sets. We can suggest that chosen has highly informative for characterizing single cell population. And now we want to approach chosen base to decipher tumor progression trajectory. And this data is sourced from the Taipei Veterans General Hospital. First, we convert the gene expression matrix into network degree matrix by using our method chosen. And then we want to define which cluster may be the tumor cell population. So we score the expression of epithelial biomarker. We can find that this orange dot here in chosen, it is more cluster and has outperformance of clustering purity score. And compared to the tumor cluster with the gene expression matrix, chosen network degree matrix can find 106 additional cell additional cell in the tumor cell population, which means chosen is able to provide a clear distinguish between tumor and non-tumor population. And then we want to focus on this tumor cluster identified for the chosen. So first, we're using slingshot to determine for subpseudotype group, and we incorporate with the concept of network flow entropy, which is the method can quantify the cell state based on the network connectivity. By analyzing the gene regulatory network and our chosen network, we aim to suggest a context-dependent role for the EMT process in HNSCC. First, just like we mentioned before, we're using slingshot to determine the first subpseudotype group. And then we hire the cytotrace to identify the differentiation direction from lower differentiation potency to higher differentiation potency state. We also score the expression of mesenchymal and cancer stem cell. We can find that the higher expression cells are located in the right group here, which means those group has higher mesenchymal and cancer stem cell characteristic. It can also validate the cytotrace to output because in biological, compared to the epithelial, mesenchymal and cancer stem cell are truly having a higher differentiation potency. So chosen trajectory inference can truly reveal the pseudotype-dependent change in mesenchymal and cancer stem cell across 4D differentiation malignant epithelial subpopulation. Here, we're using our network, we are using our chosen network in cooperate with the network flow entropy. We can see network flow entropy score is lower in the epithelial state, but as EMT progresses, the cell state shift to the mesenchymal and cancer stem cell. The network flow entropy score are gradually increases. And it also showed the chosen subnetwork illustrate change in the association among 5 partial EMT gene and cancer stem cell gene in 4 subpopulation, with the network density increases, which means the interaction between those genes are gradually enhanced. So here we can get a conclusion in this section. Our chosen network combined with a network flow entropy concept, it can truly evaluate the differentiation potency. Also, the higher of the network flow entropy score are more closer to the stem cell state. And in final analysis, by analyzing the gene regulatory network and our chosen network, we can show the change of transcription factor expression and regular activity as the pseudo time progresses. We also clustered it into the 3 ways based on the time of regulon peak. In the middle term of the heat map, reference from left plot, we emphasize the pseudotype dependent marker expression regulated from those transcription factor. We also highlight the important key and illustrated in the right side. First, we can see KL4 often occur in the epithelial state and is the key regulator of metastasis by controlling STAT3 to upregulate the CFOS expression. And the interaction between CFOS, EGR1, and JONT are also often found in the middle term of tumor progression. And the final place, MIF6 and IRX4 downstream activated gene are all related to EMT and cancer stem cell. especially the FHL1. It shows the different influence in the EMT progresses in HNSCC. So we are now cooperating with the Dr. Yang Muhua to validate our finding. So here we believe that chosen have a potential to provide a valuable biological insight from single cell network perspective. Let's move to a conclusion. Here, chosen, we use the transcriptomic correlation to lower the cell-specific network methods high false positive rate problem. And in case study, we identify for the differentiation subpopulation and link to the EMT, also combine the network flow entropy to quantify the differentiation potency. And finally, we suggest the context-dependent role for the EMT process in HNSCC. Here, chosen provide a new opportunity to study biological system at a single cell level from a network perspective. Here is the acknowledgement. Thanks for my teacher, Professor Zhu Ling, and our lab member, Ting Yi, Ting Yu, De Luan. Also, Professor Dr. Yang Muhua from Taipei Veterans General Hospital, and our sponsor. And thanks for your attention. This is my poster ID. If you want to have more discussion, you can find me there. And thanks for your attention.
Any questions online or from the audience? So for your network-based approach, the modules you identified, would it be possible to further evaluate using separate patient cohorts in the clinical setting?
You mean the FHL1?
Yeah.
We are now trying to do in the TCGA, and then if we prove it is a true finding, then we will cooperate with Yang Muhua and doing the biological validation. Yes.
Thank you for our presentation. So I think the most simple way to construct a cell-specific network is to first construct static aggregated network using all cells and delete regulations when the regulator is not expressed in the cell. So did you compare the result of your method to the simplest method? So so the findings of your method cannot be found by the simplest method.
You mean the same finding cannot be found in other single cell network method, or?
Sorry, how the English? So my question is, the findings you showed cannot be found by simply constructing cell-specific networks by deleting regulations from aggregated networks using
you mean in aggregated network, we can calculate their density, and we also can find this pattern. We can calculate the single cell in those group, and then we can combine it into the aggregated network. And in our method, we also can look at this aggregated network. We can find this pattern.
Thank you.